basically the minimal risk. So we have to go to big data or intermediate big data. So in summary, basically the new classes, IL-17 blockers, IL-23 blockers, are more than a promise for the complete clearance or almost complete clearance. Basically, there is a strong rationale in psoriasis. In psoriatic arthritis, to a lesser extent for the IL-23 inhibitors, which are not approved yet in the indication of PSA. And basically, my guess, but I don't want to anticipate on Dr. Hamada's talk because he will address what will remain from the TNF-alpha blockers. Thank you very much for your attention.
Thank you, Dr. Hervé Bachelet. You will be talking again. You will have another presentation about pustular psoriasis, the dawn of new era. Would you please start the next presentation?
Thank you so much. So basically, while psoriasis vulgaris or plaque psoriasis is a common disease, pustular psoriasis is a very rare, if not orphan disease. So the reason why we tackled this disease some years ago with some friends and colleagues from Tunisia at that time was that we were very kind of confident that working on this group of disease, we would basically identify in some family cases or isolated, sporadic cases, some new genes culprits where basically we have not a multigenic disease but a monogenic disease, one gene abnormality, one major one, and basically leading to the disease, which is a completely different approach. And hopefully these pathways might also be involved in the frequent form of the disease. So you know about the diversity of pustular psoriasis from basically probably the less rare, which is palmoplantar pustular psoriasis, but also the iconic form, which is one of the rare life-threatening forms of psoriasis, which is generalized pustular psoriasis, von Zumbusch form, or GPP. And also the acrodermatitis continua of Aleppo. These different forms might be basically associated and might associate also with psoriasis vulgaris in some patients. As a matter of fact, and to make a long story short, different groups, research groups, including ours, have been able to identify some new genes in these different settings. And basically, these genes, CARD14 is a gene which is involved in some plaque psoriasis or general pustular psoriasis forms of psoriasis, of pustular psoriasis, and basically it's a keratinocyte-associated gene. I will not talk too much about that. AP1S3 is a gene which is a little bit of cloud of mystery, interfering with the keratinocyte function, identified by Francesca Capone in UK and Jonathan Barker in some different forms of pustular psoriasis. But the iconic one is IL-36RN. IL-36RN is a gene which belongs basically to the IL-1 family. So IL-1RN is the equivalent which basically encodes the antagonist of the IL-1 receptor pathway, which you know IL-1 alpha and IL-1 beta. And basically, IL-36RN is a neighbor or a cousin, but is not overlapping regarding the function with IL-1. And IL-36 is a pro-inflammatory pathway, and when you don't have this antagonist, basically, when you have homozygous mutation of IL-36RN, so no antagonist, basically the inflammatory cascade driven by this innate immune pathway, IL-36, is completely dysregulated. And thanks to our collaboration with Slaheddin Marachi and Hamida Turki in Tunisia, in Sfax, we were able, with Asma Asma'i and collaborators, basically to identify basically what you can see on the upper left is the pedigrees of some consanguineous families from patients who are originating from Tunisia. Some were followed in our department, and the medical students had no clue. They were not, asking about basically the genetics in the families. So we did that, and then we bridged with our friends from Tunisia. And then basically having a very classical approach, we identify some what we call loss of function mutation, meaning leading to the loss of function of the protein. So the antagonist is not capable anymore to block the inflammatory cascade, which in the keratinocyte and other immune cells will drive inflammation and lead to the recruitment of neutrophils. And you can see on the bottom of this slide the different phenotypes, clinical phenotypes. All these patients are sharing the same mutation, a very severe mutation, or they don't have any antagonists. So just to tell you that basically the genes are not all about the phenotypes. So the group of Francesca Capone in the UK were able to show that basically patients, because these genetic abnormalities are found between 15% to 60% of the patients depending on the geographic territories. And that's something very interesting. So basically not all the patients are mutated for any of the three genes. But one thing is for sure is that when you have basically, when the patients are mutated for this gene, the disease onset is earlier in childhood or adolescenthood, sometimes young adults. So we don't know why. There is a kind of time lapse. And basically, there are different types of mutations. I don't want to enter into details, but what is very interesting is that, and that's from our laboratory published in 2016, in red you have the very severe mutation leading to no protein at all, and you can see maybe that what is boxed in the middle is a GPP and AGAP also, the acute generalized exanthematous pustular eruption, drug-induced supposed to be, and basically these are very severe systemic phenotypes. So the most severe is the mutation on the structure and function of the protein, the more severe is the disease, makes sense. And for the blue one, it's more likely to lead to basically localized forms of the disease. So the nice thing is that basically we don't know how much, these mutations are not found all in all in patients with plaque psoriasis. But the pathway, the pro-inflammatory agonists of the IL-36, alpha, beta, and gamma, are upregulated in psoriatic plaques. So by studying the rare variants, you might identify and decipher some mechanisms operating in the frequent forms of the disease. And that's something that we should think about through collaborations. So yes, pustular psoriasis do belong to the group of autoinflammatory syndrome, genetically inherited monogenic disease of the innate immune system, not only the skin one, but the systemic one. And basically, what are the consequences on treatment? Well, so far we know that for palmoplantar pustulosis, cyclosporine and retinoids are probably the most effective drugs. I mean, the best evidence is for cyclosporine. 2 randomized controlled trials at 1 and 2.5 milligrams per kilo and per day. These are old studies and with global score from the physician, but that said, I'm sure that this is basically some good experience and also for acitretin. As a matter of fact, it was tempting to copy and paste what was operating in plaque psoriasis vulgaris. It's quite disappointing at the moment. We know that TNF-alpha blockers are not good in this indication, palmoplantar pustulosis, and this is the randomized controlled trial, secukinumab, 2 different dosing, 300 milligrams, 150 milligrams, sub-Q versus placebo, and just look at the left hand side. You can see that only basically 26.6% of the patients are able to reach the PP PASI, so palmoplantar pustular psoriasis PASI for the palms and soles at week 16 versus basically 14.1% for the placebo, which is higher than so it's quite disappointing. Yes, it's higher at week 52, but it's open label. So it's quite disappointing. So there are big hopes coming from basically the generalized pustular psoriasis model that might apply for the palmoplantar one. We know that in GPP, all in all, even though in Japan, for instance, the IL-17 blockers have been approved, it's based on some studies which are open-labeled, and so it's difficult to interpret. And a big hope is that blocking the IL-36 receptor with a monoclonal antibody might lead basically to a major improvement. Is this the case? Well, the results from the phase 1 studies that basically Boehringer Ingelheim coordinated, and I had the chance to basically coordinate these studies with friends and colleagues from all over the globe, including our Tunisian colleagues. And then basically you can see, this is in press in the New England Journal of Medicine, that basically the proportion of patients, 7 patients, that's not a lot, one single intravenous infusion of anti-IL-36 receptor, basically almost all the patients reached the complete clearance basically as early as basically week 1. You have to be very demanding regarding time points because the disease is self-remitting. And this is an example in one patient in the upper box, which the patient is mutated. On the bottom is not another patient who is not mutated. And you can see basically the level of improvement at week 1 with a complete clearance, almost complete clearance for the mutated patients and a more delayed response for the non-mutated patient. So in summary, by tackling these rare orphan variants, we're able to identify some major inflammatory pathways which are dysregulated in the frequent forms of the disease, and by tackling, therapeutically the receptor, inhibiting the receptor, it seems to be, we have to wait for the phase 2 and the phase 3, that this is basically beneficial in pustular psoriasis. I want to acknowledge all the friends and colleagues from, of course, our Tunisian colleagues, but also our colleagues from Algeria, from other publications, from Morocco, and also the group of Jean-Laurent Casanova, Francesca Capon, and Jonathan Barker from London, UK. Thank you very much. Shukran.
Thank you, Professor Hervey, for your two interesting and informative lecture. Now we have 2 questions for the both lecture. Okay, go ahead.
The third one. So thank you for the lecture. Among the existing biologic, which one you think will help us until we get IL-36 monoclonal antibody? Among the existing biologic, which one you will use for a patient with this mutation?
Which one would I use for?
For the pustular psoriasis with IL-36 receptor mutation.
So far, with the existing one?
Yes.
It's a tough question. I would basically stay a little bit away from the biologics unless there is really, I think that infliximab in some cases, the thing is that helps. Sometimes infliximab is rapidly effective. I would say sometimes IL-17 blockers, they are rapidly effective as well. But the thing is that this should go, I think, only if cyclosporine or steroids are not, kind of effective. And I would say that basically it's unpredictable, the response. That's the thing. So IL-17, neither IL-17 nor basically TNF-alpha are the common denominators. We hope that IL-36 is the common denominator across the different genetic backgrounds, but we don't know that yet. It's suggestive, the phase 1, but we don't know that yet. Thank you. I mean, that's very important. So I would say first line infliximab and maybe second line, basically one of the IL-17 blockers like ixekizumab or secukinumab.
I have two patients with the mutation. I will discuss with you what was our success. Thank you.
Thank you. Thank you very much for your question.
Second, yeah, go ahead.
Does this gene have to do with AGEP disease as well, or didn't you study the correlation between this gene and AGEP, the drug reaction that mimics pustular psoriasis?
So AGEP has been, because I didn't catch the beginning of your question, but I guess your question is, did you tackle AGEP as a disease entity for the frequency of the mutations? The answer is other groups did. There are two levels of answers. First, in AGEP, some identical mutations compared to GPP have been found, right? So exactly the same mutation, which questions the sort of hermetic separation between AGEP and GPP. And the second reason, I think there is a challenge around the classification between the two, is that one of the